Batman vs. Owlman, the Omnivirus Pandemic. Act 1.
C4 lace to the wall. Aim for the batarang.
Laser blaster locked and loaded, waiting for your call.
Now! Diana!
These katanas should be able to cut the chains.
Ah, Batman?
Diana, you alright?
I am thirsty.
There is no water in the refrigerator, only chilled ethanol beverages which are not safe in dehydration.
Find the one with the lowest alcohol by volume.
This one got 0.5% ABV.
This will do. Nightwing, where are you? We gotta get out before anyone shows up.
Right behind you. I was grabbing some loot.
What did you find?
I got some data from a computer.
Good. Let's go.
Welcome to the Batcave.
Adjust the hybrid bed.
Ready.
Computer, start the analysis on her health.
Initiating electronic scan. Blotting any injuries. Immobilizing by anesthesia. Comparing Amazon Kryptonian hybrids. Printing radiograph. Analysis complete. No major injuries detected. Saving analysis on minor injuries.
Great. Rest here, Diana. There is no major injury. You will be fine. Richard, you too get some rest. We got a lot of work to do tomorrow.
Sure, I will sleep after decryption of the files I found in the prison. Good night.
Act 2.
Thank you for the save yesterday.
You are welcome. How is your health?
Perfect.
Put this probe on.
Is this to spy on me?
No, it is to spy on me.
Come here. What?
You might be infected. They have released a new virus. We decrypted the data that was in the prison you were in.
I hope this one is not from bats.
This one is from owls.
Hi, Richard. Thank you for the rescue.
Hello, Diana. No problem.
Hi everyone, I brought 6 handheld teleporters for our base, and here are some other new things I got. Exosuit, Tactical Saber, Tactical Biomechanical Heart, Amplified Purview Sensor, Amplified Proximity Sensor, Radiation Purview Sensor, Echolocator Claymore.
Hello, Damien. Here are your katanas. Thank you.
Hi, Damien. Use this echolocator. It is 5th generation. Remember to adjust it for your eyes.
Hello, Damien. Your katanas were really useful. Thanks. Well, everyone is here, so let's discuss the latest update.
The files called home when we cracked their encryption. It was part of their plan to leave Diana poorly guarded with files that send locations upon decryption. Now they know exactly where we are and will come here to stop me from making the cure.
This also explains why the encryption was so weak.
Get to work. We got this.
Not that simple. They have more supervillains than us. We 4 cannot do this on our own. I have made your probe radioactive and given both your bracelets a negative charge, so the probe can fit on either of them. This will give you access to my Bat computer and will allow you to communicate with us wirelessly. From the Bat computer, you all can see which superheroes are free and call them as needed.
I found this outside. It says, "Riddle me this. I start with R and end with R. I take people very far. My owners have a big home. Readers call me a palindrome. What am I?"
Race car. But what does all this mean?
They will send Race Car as their first attack.
They have sent them already.
Diana and Nightwing, search for any free superheroes. Robin, mount the Gatling cannon. If you strike a little away from the center of race cars, then you may stop them before they get too close. The roof is open, and anything can be thrown at you, so always stay in your exosuit.
Father, your 5th generation echolocator is showing blurred images. I can barely see the Gatling cannon, let alone the middle of target.
Told you to calibrate it first.
The enemies gave me no time for that. I am using my 1st generation echolocator. The length of the cars appear too short from up here. I am going for the sweet spot.
Hold on. Their armor turned out to be double of what I expected. I am doubling the firepower. Start reshooting in 3 seconds.
Wow. Thanks, both are down. One of them looks like a vector car and the other is alien to me.
Nice, Robin. Have some rest. I will take your place.
Good job, Robin. This is what I call bat defense. I will go check him out.
Act 3.
There is a man in the car. Looks like he fainted. I am taking the cars to the armory.
I am coming.
Who are you?
Good night.
Reserve power online.
Who are you?
I am Ares. I have come from the realm of darkness, and I am ready to take your soul.
Diana to the team. Ares is finished, along with all his illusions.
Act 4.
Someone dropped a disruption field generator on the roof. None of the Gatling cannons are working. Batman, can you divert emergency electricity here?
Take it. Take out that thing.
I still got no electricity. Perhaps the disruption is just too strong.
Then use the flame guns. They do not require any electrical power.
I am torching with the flame guns, but it is heat stable. In fact, it is generating a lot of heat by itself. Can you give something to cool it down?
Ice guns have been deployed. Freeze it.
Ice guns are working. Temperature is going down. They started artillery firing. It's raining death over here. Ice guns are being destroyed one by one.
Just prime 2 proximity mines on the way to the basement and get out of there. Make sure no one follows you to the basement.
Both proximity mines are primed. He is Dr. Polaris.
Aquaman, do you hear me?
Speak ahead.
Get ready for the teleportation.
Temperature is going up again. He is extending a bridge up here.
Welcome to the Batcave.
Aquaman. Nightwing is up there fighting Polaris. Everything is on fire, and looks like whatever he deployed is heat stable. If you can bring the temperature down, then that should cease all his systems. Nightwing? Nightwing! I'm going.
No, Batman. I am going. I can cool things with my water powers.
You are late, Aquaman. My work is complete.
But my work is remaining. Batman. Polaris is captured in my water chamber. I am sending you his fingerprints. Nightwing is doing fine up here. His communication system has been damaged. The artilleries are probably out of ammunition. I'm going to check out the situation.
Fingerprints matched. He is Neil. Aquaman. Send Nightwing to the computer lab to get new gadgets. I'm coming with you. Rest of you stay on Overwatch.
Act 5.
Colossus and Wolverine have completed their mission. I am calling them. Peter, Logan, do you hear me?
Yes. Say ahead.
Get ready for the teleportation.
Welcome to the Batcave. Welcome to the Batcave.
Logan, pieces of alien car are outside. Take them to the armory where you will find Vector Car's pieces for recombination. Peter, help him.
They are so sticky.
This should make your job easier.
What are you doing?
Calling Okamura.
Okamura?
Toyman. He was the one who built the first recombinant car.
Yes, Colossus. What can I do for you?
They launched a ballistic missile on the Batcave. Our defense systems have still not recovered from the effects of disruption, so I'm sending my plane to stop it in midair. But there is a chance of missing it, so you guys may abandon my Batcave.
No need to abandon. I can sit in your plane, confirm with my eyes, and tank the ballistic missile myself. Might as well save your plane.
Are you sure? His bombardment package includes tough metals like gold and tungsten.
I can take it.
Get in my plane. I will guide it remotely and eject you at the last moment. After the explosion, another plane will catch you.
Let's do it. I have visual confirmation on the target.
Get ready to jump. Opening the door in 5, 4, 3, 2, 1. Now! Gotcha.
Welcome back. That was quite a stunt you pulled off.
Thanks. Nice car. You made it so fast.
Yeah, thanks to biotechnology. Plus, I also got some help from Okamura. Diana, want to test drive?
Very good, Logan. This recombined car is perfect. It is armored with one of the toughest metals.
Cool. Then I will transform into this one.
Act 6.
Neil has been captured, but he accomplished his mission. According to what we heard and saw from him, the thing of our interest is in the basement. Now, here are the pictures he sent of the Batcave for you to study, Samuel. Summarize the plan.
I will sneak inside via amphibious combat vehicle, plant the radar jammer, identify the main transformer, and shut it down.
Good. Remember not to take the vehicle too close, or you will be detected. You will have to get out and swim when you are near their land. We will follow you in the aircraft. What are you doing, Snart?
There is a friend with similar skills. Can I call him?
No. One Captain Cold will do better than multiple Rookie Colds. Besides, we do not have time. Everyone, prepare your weapons and get in the plane.
I am near their area, leaving the vehicle and going for the radar. That idiot Aquaman left some fishes on patrol here.
What? You all right, Mirror Master?
I am fine, Riddler. Thanks to the aquatic harpoon I found in the vehicle. Radar jammer is installed. You can fly here safely now. Looking for the main transformer? Transformer is down. Defense system should stay offline for a while. You guys can paradrop now.
Team, they just took down the artilleries. It will take the Princess, Batman and Robin 322 seconds to come here. Plastique, we gotta quickly break through the entrance. You got that bomb ready?
Yup. The bomb has been planted. Initiating explosion in 5, 4, 3, 2, 1.
Nice one. Alman, starting with you. Go get the data. Others, check every room.
Intrusion alert. They have broken through the roof. I need you all in the Batcave.
No. You all come outside from the back door at my location. We will attack together. Why so much delay, Nightwing? You got no alarm?
They probably disabled our radar and transformer on the roof. I got no warnings.
I copied the data of each of them. There is no information on the thing of our interest here, but it should be in the basement.
Act 7.
Thank you for your timing, Clock King. I will take back the leadership from here. Game over, Batman. This incendiary device will end all your hard work.
Wait, this would destroy the whole building and kill all of us, including you.
No, it will only destroy the bioreactor. Trust me. How is that possible? It should be in flames.
This one is not the stirred tank bioreactor. No oxygen in it, which means no combustion fuel for your toy.
So you shifted the bioreactor elsewhere.
Maybe next time send someone better than Neil, who can give you updated information.
We will meet again soon, Batman.
Batman to the team. Owlman escaped. Simple stirred tank bioreactor is secure. Agitator system.
Ready.
Oxygen delivery system.
Ready.
Foam control system.
Ready.
Temperature control system.
Ready.
pH control system.
Ready.
Sparge stirred tank bioreactor is online.
So you knew Neil had sent them the information?
Yes. That is why I replaced the real bioreactor in the basement with a fake one.
What was in the tank in the basement?
Just water. His incendiary device consisted of white phosphorus, which turns off underwater.
And where is the real bioreactor?
It was hidden in plain sight, in the garage. The cure is ready. Get ready for a shot, everyone.
None of these syringes work on me, Bruce. I better not convert to human form until I am healed.
Do not worry. I have micro-injection technology for your thick skin. Diana, help him.
Come here, Peter. This micropipette will do.
Act 8.
Thanks to everyone. The time has come to say goodbye.
So is this vaccine ready for sale?
No, downstream processing is left.
Down what?
Downstream processing. It includes separation, purification, and preservation.
After that, we will need you guys for marketing.
Okay, no problem.
We will handle it.
Sure, anytime.
Good night.
Come here. What now?
The one who does not respect biology is not respected by biology. It will take us 6 weeks to get complete immunity.
I am an Amazon.
This is the omnivirus pandemic. Owlman wanted to end all life, not just humans. Besides, our mission is not over yet. He took away some biotechnology from here.
What part he took?
The part used to make an animal superior by introducing some foreign genes in it, changing its genotype.
The genetic engineering part.
Yes, this time we need to be ready before he unleashes his attack.
Biotechnology, principles, and processes. Alright, so this is the sequence that we will follow. Isolation of the genetic material, deoxyribonucleic acid, DNA, Act 1. The genetic material in majority of organisms is DNA, which is shown by Diana. DNA is enclosed in a cell, which is shown by prison cell. Recombinant DNA technology involves several steps, starting with isolation of DNA. This step is portrayed in the story by Diana being held captive in Owlman's prison cell and eventually being freed from that cell. Now, in order to acquire DNA, we have to break the cell open, which is achieved by different enzymes depending on the cell type. In the story, we can see that Batman and Nightwing come to rescue Diana, and every weapon that they use to destroy something accurately reflects the combination of the enzyme and its substrate. So lysozyme digesting cell membrane of bacteria is shown by laser blaster shooting at Batarang. Cellulase digesting cell wall of plants is shown by composition 4 bomb demolishing the prison wall. and chitinase diastine chitin of fungus is shown by katanas cutting the chain. Breaking the cell open is not enough to get pure DNA. We also need to digest other molecules like ribonucleic acid by ribonuclease and proteins by protease. Finally, DNA precipitates out by addition of chilled ethanol, which is shown by Batman giving a cold alcoholic drink to Diana. Act 1 was supposed to end here, but there is a random question on sudden hybridization in your past papers, so I had to include it for completion. Sudden hybridization, which is shown by hybrid bed, is a method used for detection and quantification of a specific DNA sequence in DNA samples. It involves electrophoresis, shown by electronic scan, blotting, shown by blotting of injuries, immobilization, shown by immobilizing by anesthesia, hybridization, shown by Amazon Kryptonian hybrids comparison, and autoradiography, shown by radiograph. Cutting of DNA at specific locations by restriction enzymes. Act 2. The DNA we isolated in previous step needs to be cut by the enzyme called restriction endonuclease, which is shown by Robin. The first type 2 restriction enzyme isolated was HindD2, which is shown by handheld teleporter, and it is the first item that Robin mentions. It recognizes a palindromic nucleotide sequence of 6 base pairs, which is shown by Robin bringing 6 teleporters for the base. The plasmid PBR322 has restriction sites on different genes for different restrictases. On the ampicillin resistance gene of the plasmid, we have sites for PST1 and PVU1, which are shown by amplified proximity sensor and amplified purview sensor. On the tetracycline resistance gene of the plasmid, we have sites for BAMH1 and SAL1, which are shown by tactical biomechanical heart and tactical saber. On the repressor of primer gene of the plasmid, we have site for PVU2, which is shown by radiation purview sensor. On other genes of the plasmid, we have sites for EcoR1, CLA1, and HindD3, which are shown by first generation echolocator, Claymore, and handheld teleporter. Other examples of restrictases include XHO1 and EcoR5, which are shown by Exosuit and 5th generation echolocator. EcoR5 produces blunt ends on their substrate. This is depicted in the story when Batman gives Robin his 5th generation echolocator, and that ends up producing blurred images. Now, before we study how restriction enzymes work, we need to discuss about palindromes. In general, a palindrome is a word that reads the same backwards as forwards, such as civic or race car. In genetics, a palindrome is a nucleic acid sequence in a double-stranded DNA or RNA, whereby reading in a certain direction on one strand is identical to reading in the same direction on the complementary strand. Didn't understand it? Never mind, you will once you start solving the upcoming questions. So, the method I use to identify palindromes is to draw a line vertically through the center of a strand and fold it along this line. If the bases pair, that is guanine pairs with cytosine and adenine pairs with thymine or uracil, then it is a palindromic nucleotide sequence. That is all you need to do if you are given only one strand. When you are given 2 strands, however, you will need to read the second strand in the same direction and check if it is complementary and exactly matches the first strand. Pause the video here and try to solve which of these 12 are palindromes. Okay. Note that some questions trick you with directions. Like question 11, if you ignore directions, looks like a perfect palindrome. But if you read both strands in 3' to 5' direction, then you will immediately notice that the starting bases, thymine and adenine, as well as the rest of the bases do not match at all. Similarly, in question 7, let's say you want to read both strands in 5' to 3' direction, then neither the starting bases, guanine and cytosine, nor the rest of them match. Phew, that was a huge ride on race cars. I mean palindromes. Now, I do not want to break your heart, but even after perfecting the palindrome concept, you might get a question which requires rote memorization. And this is where my story Mnemonic comes in. In the story, Robin puts on his first generation echolocator and sits on top of the Gatling turret. This is a representation of the enzyme EcoR1 being able to recognize the palindrome 5' GAATC3', 3' CTTAAG 5'. Note that Robin is sitting on top of the Gatling cannon, which means we are concerned with the top strand, which is written in 5' to 3' direction. Generally, restrictases work by identifying palindromes, which are shown as race cars, inspecting their lengths, which is mentioned as Robin inspecting the race cars from top, and then attacking their sugar phosphate backbones, which is mentioned as Robin going for the sweet spot. Restriction endonucleases sometimes cut a little away from the center, which is mentioned as Batman advising Robin to strike a little away from the center. Batman also advises Robin to stay in his exosuit, which is a reference to restriction enzymes being able to work in vitro, meaning in an artificial environment like a test tube. In the end, Batman realizes that he has to double the firepower in accordance with the double armor of the race cars. This illustrates that the restriction enzyme will cut both strands of the double-stranded DNA. After destroying the race cars, Robin recognizes one as a vector car and the other is foreign to him. This is a representation of vector DNA and foreign DNA. Also, Robin attacking both vector car and alien car demonstrates the fact that both vector DNA and alien DNA are attacked by the same restriction enzyme. Agarose Gel Electrophoresis, Isolation of DNA Fragments, Act 3. The fragments of DNA made by restrictases are separated via agarose gel electrophoresis. The agarose matrix, which is shown by arrows, provides a sieve-like effect. As DNA is already negatively charged, which is mentioned as Diana's bracelets given negative charge, we can move it towards anode terminal under an electric field through the agarose matrix, which is shown by arrows forcing Diana towards an electric terminal. The separated DNA fragments can be visualized if stained by ethidium bromide and placed under ultraviolet radiation, which are shown by electromagnetic pulse, EMP grenade, and sun, as 10% of sunlight is UV light. The fragments appear as bright orange bands, which is shown by car pieces burning in bright orange flames. In the end, Diana says that she got rid of Ares and all his illusions. This is a reference to elution, the process of extraction of fragments from agarose gel. Amplification of gene of interest using polymerase chain reaction, Act 4. The required fragment of DNA can be amplified by polymerase chain reaction. PCR got 3 steps. Step 1. Denaturation, shown by disruption field generator. In the first step, temperature is raised to about boiling point of water, 98 degrees Celsius, which is shown by a flame gun. This causes double-stranded DNA molecule to melt or denature, yielding 2 single-stranded DNA molecules. Step 2. Annealing, shown by artillery firing. In the second step, temperature is cooled down to about half boiling point of water, 50 degrees Celsius, which is shown by ice gun. This causes annealing of single-stranded DNA molecules to primers, which are shown by 2 proximity mines. This way, 2 primer template hybrids are formed, and from there, the DNA polymerase, which is shown by Dr. Polaris, starts building DNA. Step 3. Extension or elongation, shown by bridge extension. In the third step, temperature is again raised back up, this time to about 3/4 boiling point of water, 72 degrees Celsius, which is shown as ice guns no longer functioning, which means there is nothing to keep things cool. This causes Taq polymerase to synthesize new DNA strands complementary to the template DNA strands. Using PCR, a billion copies of DNA can be made. Coming back to the story, Batman advises Aquaman to bring temperature down to stop all systems of Polaris. This is a reference to the importance of high temperature in this reaction. Also, Aquaman locking up Polaris in his water chamber is a representation of polymerase present in Thermus Aquaticus. Finally, Aquaman sending fingerprints to Batman is a depiction of PCR being used in genetic fingerprinting. Ligation and insertion of recombinant DNA into the host cell or organism. Act 5. The amplified DNA fragment can be joined to vector DNA using the DNA ligase enzyme, which is shown by Logan. If same restriction enzyme is used to cut the alien DNA and vector DNA, then the DNAs will have same kind of sticky ends, resulting in ligase working more efficiently, which is mentioned as Colossus replying to Logan that sticky car pieces will make his job easier. The end product of ligation is recombinant DNA, which is shown by recombinant car with Diana inside. And the first recombinant DNA was made using plasmid of Salmonella typhimurium, which is shown by Okamura Twyman. In genetic engineering, we have some methods through which this recombinant DNA can be inserted into host cells. One of them is called biolistics, which is shown by ballistic missile. The device is called gene gun or biologistic particle delivery system. A very useful host cell in genetic engineering experiments is Escherichia coli, which is shown by Colossus. So what happens in biolistics is that the cells are bombarded with particles of gold and tungsten coated with recombinant DNA, which is mentioned as Batman warning Colossus that bombardment package consists of gold and tungsten. After bombardment, recombinant DNA enters the cells and transforms them, which is shown by Colossus transforming into a tougher state after touching recombinant car with Diana. Another method called viral transformation uses disarmed pathogen vectors, which is shown in Act 7 by Diana disarming Clock King. When they are allowed to infect the cell, they transfer the recombinant DNA into the host. One more method called microinjection uses micropipette to inject recombinant DNA directly into the nucleus of an animal cell, which is shown in Act 7 by Diana holding micropipette for Colossus. Cloning vectors, Act 6. Cloning vector, which is shown by Clock King, is a piece of DNA into which a foreign DNA fragment can be inserted for cloning. The features of a cloning vector include 1. Origin of replication, shown by Owlman. 2. Cloning site, shown by Captain Snart. 3. Selectable and screenable marker, shown by Mirror Master and Radar Jammer. 4. Repressor of primer, shown by Riddler. 1. Replication origin is the sequence from where replication starts, which is mentioned as Clock King asking Owlman to start entering the Batcave. It also controls the copy number of the linked DNA, which is shown by Owlman copying files about Diana. 2. Cloning site is restriction enzyme recognition site we talked about earlier. To avoid generation of several fragments, presence of only one restriction site is preferred, which is mentioned as Clock King refusing the request of Captain Cold to bring in his friend. 3. A marker gene is a gene used to determine if a nucleic acid sequence has been successfully inserted into an organism's DNA. It has two types. Type 1, selectable marker gene. It is used to identify transformants and non-transformants by killing one of the groups of cells, which is shown by Mirror Master identifying the main transformer and shutting it down. Selectable markers are often antibiotic resistance genes, which is shown by aquatic harpoon. For example, ampicillin resistant gene, which is shown by amphibious combat vehicle. Type 2, screenable marker gene, also known as reporter gene, which is shown by Radar Jammer. It is used to measure the level of expression of the transformed gene. The types of cloning vector include 1. Bacterial artificial chromosome, shown by barrel, scattergun or shotgun, and backpack. 2. Yeast artificial chromosome, shown by octosecond timer. 3. Retrovirus, shown by retractable knife. It is used as a vector for introducing a DNA fragment in human lymphocytes, which is shown by laser sight. 4. Plasmid, shown by plastique. It is double-stranded and circular in structure, which is shown by plastique holding two circular bombs. It can clone only a small fragment of DNA, which is shown by fragmentation grenade. It can join eukaryotic DNA, which is shown by electromagnetic pulse grenade. Now, there are two types of plasmids. Type 1, plasmids that are said to be under relaxed control. In these, the initiation of replication is controlled by plasmid gene, which is mentioned as plastique initiating the bomb explosion. Type 2, plasmids that are said to be under stringent control. In these, the whole process of replication, including initiation, is controlled by bacterial gene. Tumor-inducing plasmid, which is shown by titanium necklace, is often used for making transgenic plants. It is found in Agrobacterium tumefaciens, which is shown by titanium flashlight. Plasmids can also be artificially created. A popular example is PBR 322, which is mentioned as Princess, Batman, and Robin coming in 322 seconds. Some cloning vectors are pathogenic and they must be disarmed before they are allowed to infect the host cell and transfer recombinant DNA into the host. They are the same disarmed pathogen vectors mentioned earlier. Obtaining the foreign gene product. Act 7. In almost all recombinant technologies, the aim is to produce a desirable protein by the expression of recombinant DNA. In the story, the product is a synthetic vaccine made by Batman. A bioreactor provides the optimal growth conditions for achieving the desired product. The most commonly used bioreactors are of stirring type, which include simple stirred tank bioreactor and sparged stirred tank bioreactor. Stirred tank bioreactor is designed for the availability of oxygen throughout the process. This is clearly emphasized in the story. Downstream Processing, Act 8. After completion of the biosynthetic stage, the product has to be subjected through the processes of separation and purification, collectively referred to as downstream processing. Then it is ready for marketing. That is all. Go seek discomfort and disconfirming evidences. Assalamu alaikum.